Summarize the whole HMP shunt. HMP shunt, two functions are there. One is to produce NADPH, second is to produce pentose phosphate, pentose sugar. You all know pentose sugars are required. Pentose sugars are required for nucleotide. DNA, RNA, you all know they have nucleotide, they are having sugar, pentose sugar. So pentose sugar are coming from HMP shunt. So HMP shunt is producing pentose sugar, that's why we say pentose phosphate pathway. So you can divide HMP shunt in two fragments. One is oxidative phase, second is non-oxidative phase. Oxidative phase is irreversible, non-oxidative phase is reversible, and we found that oxidative phase means hydrogen is removed. Oxidative phase means hydrogen is removed and hydrogen is taken up by NADP to form NADPH. So oxidative phase produce NADPH. This is irreversible, and in this the main enzyme, rate limiting enzyme, is glucose 6-phosphate dehydrogenase. Then non-oxidative phase, in this pentose sugars are produced. Okay, we found that this NADPH has multiple role in our body. Yes, NADPH is used in fatty acid synthesis, cholesterol synthesis, steroid hormone synthesis, sex hormone synthesis, to convert oxidized glutathione to reduced glutathione in RBC. So these are the common site, common site of HMP shunt. Common sites of HMP shunt. Okay, this NADPH is produced by glucose 6-phosphate dehydrogenase. Whenever dehydrogenase is there, hydrogen is removed. You all know, whenever dehydrogenase is there, hydrogen is removed. Hydrogen is taken up either by NAD, FAD or NADP. Here the hydrogen is taken by NADP to form NADPH. Clear? Now, yes, you can, if you want to remember, yes, I can give you what is the role of NADPH in RBC? What is the role of NADPH in RBC? Yes, in RBC there are reduced glutathione. Reduced glutathione, I can write like this: GSH 2 molecule, GSH 2 molecule. These two molecules may join to form GSSG. So you will remember that in complete biochemistry, whenever sulfur would be there, it will make disulfide bond and 2 hydrogen will be released. 2 hydrogen may bind with H2O2. When H2O2 bind with 2 hydrogen, they will form 2 molecules of H2O. Clear? So you can easily remember, yes, that H2O2 bind with 2H means H2O2 is toxic. This H2O2 can oxidize hemoglobin. What I said, H2O2 can oxidize hemoglobin. Okay, but if normally this reduced glutathione is present, this convert H2O2 to water. So this detoxify H2O2 by enzyme glutathione peroxidase. Glutathione peroxidase. This is 2H2O, but it is oxidized. So this is oxidized glutathione. This oxidized glutathione can be converted back to reduced form with the help of glutathione reductase. And glutathione reductase, the cofactor is FAD. The cofactor is FAD. FAD means vitamin B2. So in this reduction, wherever you want reduction, you require NADPH. So this NADPH comes from HMP shunt. So if this enzyme is deficient, this NADPH would not be there. If NADPH would not be there, this oxidized glutathione will not be converted back to reduced form. And when further H2O2 would come, further oxidative stress will come, then it will produce hemolysis by oxidizing hemoglobin, methemoglobin, hinge bodies, yes, and will precipitate on the cell membrane, RBC membrane. Macrophage will come and will bite this hinge body, and this will be called at bite cell. So hemolysis would be there, bite cell would be there. Clear? In G6PD deficiency, you must have read in detail with the images also in pathology.